morning is Eric Smith, who's right over in the corner, and he is going to be talking about combinatorial chemistry with rule-based systems and its associated information theory. Eric.
Okay, thank you, Lynn. Thank you. Especially to the organizers for what's been a really fun and interesting meeting. I think I've been able to learn a lot. I think I should preface my comments this morning with a couple points of history to set things in context. One point is my own history of work in this area. For about the last 20 years, my colleagues and I have been trying to ask chance of necessity questions about the origin of life. A lot of this has centered on metabolism, and it's been descriptive in character, looking at features in metabolic reactions, network relations, their place in physiology, the genetic code, etc., that are functionally important, and it seems surprising to be sort of all carried by the same small number of components. And a little bit of that is okay, maybe it makes a contribution, but ultimately, questions of chance and necessity are questions about counterfactuals. And because we did not have good tools for elaborating and reasoning quantitatively about counterfactuals, at some point we were not contributing anything more. So today's talk is in one part an effort to begin to build some of those tools. Second point of history is the history of science in the 20th century. There were three important revolutions in physics in the 20th century, important because they changed our entire concept structure for understanding the world. Of the three of them, the one that's most important for all of science and inadequately communicated in terms of creating not only a theory of objects and forces, but of causation, information, and robustness, is thermodynamics. Now, in spite of the fact that it took us more than a century to get clear what we were doing in thermodynamics, all of it was worked out for systems that fundamentally are simple. They have a lot of symmetry, they have a lot of redundancy, they lack intrinsic complexity in the state space, which characterizes chemical reaction systems, especially when they're driven away from equilibrium. So the second point of this work is to try to understand what thermodynamic reasoning gives us in the way of concept structure for the kinds of complex spaces that actually host the questions we care about for counterfactuals. Now, this is going to be a super short talk. All I can try to do is give you a flavor of the way we would set up and work through one of these problems, the kinds of tools we use, and the kinds of calculations we can do with it. I do want to comment that the work I'll show you is a kind of a tempo andante collaboration over the past several years with Jakob Andersen at the University of Southern Denmark and Harrison Smith at Helsinki. So, I'll cover 5 topics, but really there are 2 main ideas. The first one is approaching chemical reaction systems as what are called rule-based systems, or 3-level systems. The point here is that constraint and complexity can enter systems like this at multiple levels, and we want an abstraction that allows us to derive consequences within and between each of the rules directly, or each of the levels directly. Second thing I'll show you is I'll set up the little toy problem that we use as a working example in this talk, which is the question of counterfactuals to the observed universality of sugar-phosphate biochemistry. And in particular, what kind of an evolutionary problem that is, and what information and work are associated with the universality we see. There will be a little digression where I'll just show you what it means to derive something directly from rules in order to bypass sort of exhaustive enumeration of instances, which is what we often do when we use computers to handle complexity, and in particular, how this can allow us to recognize a kind of algorithmic structure in the way sugar-phosphate chemistry is actually solving the problem it solves in biology. Fourth point is the second main idea, and that is, how do we recognize the information that is inherent in these kinds of questions about evolution? And then depending on how we set the problem up, we find in some cases that the proper measures of information are actually related to functions of thermodynamic state variables of a kind that are familiar, though requiring some justification to argue that they belong there. And then the last thing I'm going to bring in, because I think it's beautiful and clarifying, the informations in which these problems live actually have a geometry. And when we recognize that there is a geometry underneath of it, in addition to allowing us to see what's going on, it allows us to recognize that these problems sometimes decompose. So we don't need to know all details about everything to compute correct answers for some subset of the problem. So, what am I calling rule-based or 3-level systems? The idea is that in something like stochastic chemical reaction systems, there are actually 3 levels of abstraction we can recognize. First, oh boy, this is really blanched, okay. First level are what I'm calling rules. These are basically chemical reaction mechanisms. These operate at the level of atomic centers, bonds that are rearranged, functional groups, and so forth, not whole molecules. But it is in the rules that patterns are created or consumed. Now, the rules are then embedded in literal molecules that undergo literal reactions. And so the second level in a 3-level system is what we would call the reaction, the stochastic chemical reaction network. Stochastic or deterministic or otherwise. The third level is the actual dynamics of states, their trajectories or the probability distributions on them in state spaces. So at the rule level, the relevant mathematics is the dependency among the creation or consumption or use of patterns. At the reaction network level, the relevant mathematics is the mathematics of hypergraphs, which turns out to be richer and more expressive than simple networks, but also computationally more complex. And then, at the domain of state spaces, we have mastery equations, the dynamics of probability distributions, and stochastic trajectories. One thing I want to emphasize is that each of these levels is a generator of regularity in the levels beneath it, and a finite system at one level can generate an indefinitely large system carrying that regularity at a lower level. It's useful to notice this not only because it makes these systems comprehensible to us, but because it gets at why a system that looks very messy or structurally complicated may have been amenable to the incorporation of information, first in geochemistry, and then later may have posed solvable selection problems to evolution. So I want to note just in passing that the examples I'll show you were generated with a graph rewriting abstraction for computational chemistry. It's called NUD. It's maintained at the University of Southern Denmark by Jakob Andersen. And this is an open source code base, online sandbox where you can play, list of references, and lots of great stuff to dig in to understand what graph rewriting systems do for chemistry as a rule-based system. So the way we use a rule-based system to get from an encoding of our knowledge of mechanism to dynamics is meant to mimic what chemists are already doing, though, of course, what I'll show you is at very, very limited simple levels. A rule is basically a collection of atomic centers and bonds that are rearranged in going from edots to products, together with a requirement that these can be embedded in an order-preserving way in explicit molecules. And so the context in the rule level comes from a combination of what is created in going from edot to product and in what is produced by the way this rule is embedded in the particular molecule. In order to get from that embedding to a reaction network, we can start with a seed molecule, and then we can recursively apply this embedding anywhere we can find an instantiation of one of the rules in the molecules we have to produce further molecules, and in this way open the network by what's called network expansion. So having told you that that's going to be our platform, let me introduce you to the evolutionary puzzle that will be our example. Sugar chemistry is famously combinatorially complex. We saw earlier in the week the sort of monumental paper by Hook's group laying out the big tangle of the foremost network. But if we look at biochemistry, there is essentially a universal pathway that does the reconfiguration from 3 to 5 carbon sugars. It is almost invariant and is one of the oldest pathways in all of biochemistry, instantiated in a way that you can't see here in the bleach, in a catabolic direction, the pentose phosphate pathway, and nearly identical as part of a carbon fixation cycle in calopepsin. So, we want to understand something, and I've now confused myself and try to remember what we want to understand. Yeah. What we want to understand is this universality a frozen accident, or are these pathways chosen because they're optimal for something? If so, for what? And then what is the evolutionary work both how much and of what kind, to get to this universality from a network which at the mechanism level is sort of tends to be very promiscuous because the mechanisms can be instantiated everywhere. So to capture this as a rule-based problem, we recognize that biological sugar phosphate chemistry is really generated by 5 rules. And this is from a paper by Anderson et al. Encoding these as graph rewriting procedures. So we have aldol condensation between an aldose monophosphate and a ketose monophosphate to produce a bisphosphate. We have aldose to ketose conversions going either way. We have hydrolysis of phosphates from bisphosphates to leave ketose phosphates. And then we have 2 shuffling reactions, transaldolase and transketolase, that move respectively the C3 end or the C2 end of a ketose phosphate sugar. And it is using these that the universal networks of biochemical sugar phosphate chemistry can overcome the problem of no common divisors in the small integers of sugars while losing no carbon from the system, and in the case of Calvin also being quite conservative with energy. So if I apply these rules, I can then generate networks, and I will display the networks to you in this fashion. So this is kind of a subway map to show you what the diagrammatic means. The chemical species, I'll make colored dots, and I'll just arrange them around the circumference of the plot, and I'll put them in quadrants that lay things out in kind of a neat way. So the aldose monophosphates are over here, the ketose monophosphates are here, the bisphosphates are here, I'll put water here, and orthophosphate here, and the numbers around the outside tell you how many carbons there are in each of the sugars. Then, if I lay all of the reactions that can be used in the conversion that we'll study, I get a network that looks like this. The legend here for hypergraphs is the following. The species are what we said. A reaction is this solid line. You can think of this as going through the transition state if you're a physical chemist. And then very importantly, we recognize 2 kinds of nodes, which in the language of Martin Feinberg and Horn and Jackson are called complexes. These are the particular constellations of input or output molecules that undergo a reaction. Then the thin lines here are the stoichiometry that tell you how much of which species make up a given complex. Now, this network is a little finite segment. It extends to infinity. Of course, it isn't really very complex compared to the complexity you can get from all of sugar chemistry. But the point I want to make is that it contains the kind of state space complexity that we have never used thermodynamics to understand in the physical theory of matter. So this is where we want to extend the search for concepts into a place where they're underdeveloped, but which is particularly, I would say, an area of opportunity for the physics of living systems. So the network is the possibility space for conversions. If I tell you I want to make a given conversion, like converting 5 3-carbon sugars to 3 5-carbon sugars using water to carry off the needed phosphate, or bring it in, carry it off, and then lose nothing else. A pathway is an assignment of how many firings of each reaction will do precisely this in this network and nothing else. So this is a problem in interlinear programming. We can hand it to the computer. The computer will give us back solutions. And of course, early in the list, it finds the biological solutions. A thing that I found surprising is that in this little tiny network, we have 763 distinct solutions before we bothered to stop enumerating, because we figured this was enough for me to study and find structure, even if I'm very stupid. But this gives you a flavor of what the selection problem is as a needle in the haystack problem. There's a tremendous diversity of possible solutions, and biochemistry is using a particular one. We want to understand what that work of pruning is. So, if we see that a pathway is universal, for what might it be universal? Well, here I'm showing you 2 properties. This is just an index list of the 800 with the duplicates removed. And down here I show you how many distinct kinds of reactions are used. That might be a kind of a cost criterion. How many genes do you have to maintain and transcribe and so forth. And then here, this is a measure of the chemical work needed to force a given unit of conversion, or the topological contribution to that chemical work. I'll say more about it as we come toward the end. And what you see right here is that the Calvin stands out singularly as a minimum of that. So we would like to understand whether, where that comes comes from and what the selection problem is to get to it, and also sort of what it means. So, digression. What does having a rule-based approach add to just brute force modeling? In particular, can we prove that the minimality I just showed you is a property of the rules, rather than just witnessing it in what a computer gives us back? There's tons more we can do, but today I can't cover it. So, interesting thing about rules. Of the set that I showed you, all of the complexes are either a single sugar or a pair of sugars. 5 left? Wow, trouble, okay. That means I can graph them as points in a state space, and every rule acts on one of these points by some symmetry. Oh goodness. In 5 minutes, I'm going to skip this, because there's something I want to tell you more than I want to tell you this. But we can make lovely diagrams. We can show that every possible solution to the problem turns out to be a closed curve, and there are only 2 ways to get a closed curve that only uses 7 reactions. Offline, I will tell you that story. But what I really want to tell you, because this was a conversation that came up following Kate's talk, where Steve asked for something that really, really does belong, is what is the information in a pathway and the cost to select it? So what does it mean to prune from a network enabled by mechanism to a pathway? And why must it have a cost? Where does that cost come from? So I'm going to go after this the way we go after it in thermodynamics. I first have to tell you that thermodynamics is about costs, and what they are costs in is likelihood. Remember, I did not say energy. So, what have we been doing for 130 years in thermodynamics? Basically this, with variations. You take the gray curve here, which is some combinatorials, like how many ways you can make a certain macro state. And then you say, what happens if I bias the sampling, let's say with a Boltzmann factor, so that some states are easier to sample than others? I can create a generating function, and I can create a most likely state that's different from the state under just the combinatorics. But then what I want to know is actually what part of that tilted distribution is the measure in the underlying distribution. How likely is it as a large deviation from the background? And for that we use a Legendre transform. And from there, this is the origin of the entropy. That was for a sample from a single distribution at one time. But now let me just tell you, okay, these distributions evolve in time. Do we care? Don't know, but we can describe it. If there's a master equation that tells you how probability flows among states, then this Legendre transform of the generating function also evolves under a generator that we can derive. And in fact, we can write this entropy that I showed you here as a kind of an integral along a stationary trajectory, which says the most likely path may have been this one. What is the cost and likelihood to choose some other path? It's just this integral along that trajectory. And for any chemical reaction network with mass action, we can write down the form directly. So in fact, the accumulation of unlikelihood here, we now write as an accumulation of unlikelihood over time. Okay, it looks like we've added complexity but not done anything. But it turns out now we can do something surprising. We have an extended time distribution of what a background would do just sitting there and not changing. We can now ask, what is the likelihood for a system to persistently take a trajectory different from the most likely one? Well, we get that the same way as we got for the single time, except that now we can put a kind of attraction on the reactions and say, how hard would you have to drag reactions to displace the currents? We have an entropy function that's an integral, the same as we had before, and in fact we can reduce it. This thing, which I'll show you in red in the next several slides, is the measure of how much likelihood you have to deprive a system of to get a trajectory other than the native one. So how do we want to apply this to our problem? Well, there is only one world, and it's a stochastic world. Our network is a part of it. That world contains many other things. It contains non-specific catalysts, which may open all the mechanisms, but not select species. But it also contains cells and evolutionary population processes, and there are specific catalysts that may amplify one reaction or suppress others. We want to know what is most likely in the world, including our system, which may not be what's most likely in the system itself, because we may need to drive it. What is most likely in the world will occur where the balance of likelihood that we deny the system is exactly equaled by the balance that it gives to the world around it to do something with its products and the free energy that it doesn't waste. So, what does the construction I showed you before have to do with this problem? Well, the idea is that David was there in the marble for millennia. What we needed the sculptor to do was remove the unneeded parts of the marble to reveal him. So, likewise here, the mechanism part introduces the entire network. In order to get a measure of the cost for a particular pathway, we recognize that given any solution, like the unstructured diffusion through the whole network, any particular pathway differs from it by just a null solution that doesn't change the throughput, but that can be adjusted away from mass action to turn off the use of certain reactions, after which we can remove them. Remarkably, the cost of doing that turns out to just separate into a sum of two parts. One of which is the cost of just the likelihood deprived by driving something through the network. The second is the cost of pruning to a particular pathway. The fact that these add is what allows us to compare one evolutionary solution to another as the difference of these things, and it has just the same form of an accumulation of unlikelihood that we have for the single-time problem. Now, it is known that this kind of a separation, these are a kind of relative entropy, they're a nonlinear function. Not so obvious that they should partition into a sum. When this happens, it is actually because they are orthogonal in a native geometry of this information. I have a bunch of slides to show you what that geometry is, and I have used my time without getting to them. So, if you want to know what's on these slides, let's talk about it afterward. But I want to leave you with these thoughts. Graph rewriting systems, which were initially very naive, have matured to the point where they really are a multifunctional addition to what we can do in computational systems chemistry. Not everything, but more than nothing. Rule algebras give us the ability to do global analyses from traditional physics concepts like symmetry and conservation for whole network properties without enumeration. I really want you to appreciate that thermodynamics was always about large deviation theory, and large deviation theory is not limited to energy. It can organize a great many problems of unlikelihood in the domain of large numbers that are very pertinent to the kinds of questions we want to understand here. And it's wonderful that these information problems have geometries in which we can recognize decomposition by kinds of orthogonality, and we can see what evolution is doing as tracing geodesics in these information geometries the way you might plot an international flight on a globe. So, great thanks for your attention, and my thanks to the people who support the work.
Great, thank you so much. Wonderful talk. This is Savis from University of Utah. So this is all fantastic. How do you bring space into it?
Wonderful. Yeah, good. As with anything, you can do so in multiple ways. In flux balance modeling, which is often used for metabolic balance in cellular systems, one way you can bring space is by recognizing that a metabolite in two places is two different metabolites and the exchange is a reaction. But you can also recognize that things like membrane chemical potential are inherently geometric and topological, and that's not a graph theoretic kind of math, but it is certainly a kind of math that we have ways to represent. So you decide what problem you want to ask, what probability distribution encodes the framing of that problem and its answer, and then you figure out what combination of topological spaces, metric spaces, whatever else you wish to use to properly capture your commitment to what the problem is. But this is in some sense just the kind of ordinary scientific work that we're already good at doing. So it's just a matter of work and individual problem solving.
Paul Robert, University of Cambridge. Thank you for an absolutely fascinating talk. It's helping me think about ways that I might be able to understand whether the networks that I'm looking at are actually emerging into something. One thing I was very curious about, some of the mathematical formalism looks a lot like minimizing the action. It looks a lot like Lagrangian mechanics. And one of the features of Lagrangian mechanics is it's really important to choose the starting and the end point, and we can look at all the paths in between.
Yes.
How much of your analysis is affected or maybe even biased by your choice of both starting and ending points?
All of it and none of it. So this is the most incredible and wonderful thing. We have irreversible processes. There's not a conservation of trajectories in space. And yet, in the probability domain, we wind up getting a Hamiltonian system and a whole Hamilton-Jacobi theory. It turns out that the conserved volume inherent in that is a volume that is in part the differentiation of the process and in part the differentiation of our inference, what we wish to ask or how we wish to tilt. And so in fact, these stationary trajectories on the Hamilton-Jacobi theory, this is what's called an Eichenal approximation to, it's the leading exponential order of approximation to the time distribution of any probability distribution. A non-stationary one, a stationary one, from any initial condition to any point you wish to interrogate. Now, these are not always free of caustics. They don't always cover the whole state space. This is all the usual problems of dynamical systems. But these are problems that are well known, and we know how, what it means to encounter them. We have a whole variety of expert techniques for trying to get past them one way or another when they come up. But this is the lovely thing. Once you're in Hamilton-Jacobi world, it's like you have been given classical mechanics all over for the second time.
That part's really attractive.
Yes.
Yeah.
I'll go ahead and ask a question then. I must say I was very concerned about your talk up to about 5, 6 slides from the end, when you brought in all the evolution parts. And I must say I'm still very influenced by Norm Horowitz's 1940, whatever it was, paper, on the evolution of metabolic pathways being backwards and so on. And so I'm wondering whether what you're really showing in the end is that delta between what actually has happened in the world. So in other words, the other part of the equation is the power of natural selection of what is already there, Norm Horowitz and so on, because your sum is let's just pick a number 1. So you're showing how much importance there is to the selective aspect, to the pre-existing, the exaptation and so on, versus what would happen in an ideal world based on thermodynamics. Does that make sense?
I don't think that's what I'm doing now.
Okay. Okay, time's up.
Yeah.
Please.
So there's a kind of a physical material way we can say this that makes it seem actually completely obvious. If I have 2 ways of performing something, one of which costs a lot more chemical energy to drive than another, a lot more chemical work to drive than another, because we have to go by some circuitous route, that chemical energy is not available to do something else the cell would like to do. So that's going to show up as a fitness cost. If other things were equal, then taking a path of less resistance to do the same job puts a cell in a better position for all of its other concerns than taking a path of more resistance. It is not at all obvious that a thermodynamic variable should be the appropriate measure of cost for a cellular population process that is subject to historical contingency and all sorts of other things, which may make thermodynamics true, but not the relevant binding constraint. The work of deciding when it is or is not relevant actually falls on you in framing what your commitment is to the nature of the problem. And so just these tools will never do that for you. That's a thing where you have the liberty to make your own mistakes. But there's no sense of an ideal world. The sense is, it puts you on the line to state what your null model is. And then to say within that null model, how special is a particular thing out of the spectrum of possibilities. Because what we mean by information is whatever stipulates that particularity out of the generality. And then we want to find out, in the probability that you have said describes your model of the world, what is the likelihood cost to get to that thing that has to be paid by something else? And then you can go on to try to find the something else that's paying it. So it's really free of prior prejudices. It's meant to be a computation tool and a tool for organizing problems.
Therefore, the inefficiencies. Anyway, Are there other questions? I don't want to make it more time. Oh,
I'm just trying.
So at the end of the day, I mean, the rule-based thing is something different, because that's used that as a generative way to generate your network.
Yes.
But by the time you're left with a chemical reaction network, your procedure, aside from the mathematics, which is complex, but is essentially you look at different paths and compare them by, in a sense, how much energy you have to waste, because you climb big hills and then you go back down and all that energy gets wasted. So
For this problem, yes.
yes, for this problem, no,
Yes.
for this problem. So you have to make an assumption that, as I think you said, which is that is the driving force for how the system evolves.
Yep.
But I can think of many other driving forces. I can say, you know, you're generating all these intermediate species as part of what you care about, and some of those intermediate species are deadly for the cell. They kill other parts of the cell. So no matter how efficient they are, you would avoid that like the plague. It is a plague.
Totally.
So I don't quite understand how you figure that out as part of this problem. I understand how you make the assumption of how to optimize things and then use the algebra to figure out how that optimization works. But I don't understand how you take into account all the possible other things the cell might be trying to optimize.
Yeah, neither does anyone else. Not you or me or anyone, right? Because that's the infinite rest of doing biology and doing origin of life. And so that was not something that I was claiming to contribute anything to, because there's an infinite amount of work in that. And that is to be done basically ad hoc to the case in hand. Because I can think of a zillion other things too that I can't calculate and that I can't show you, and I couldn't show you this in 20 minutes. So I chose the simplest thing, which was, the reason we did shorter phosphates is that they're mechanistically very flat. There's not a lot of structure and heterogeneity in the mechanism, and yet they generate enough combinatorics that there's something I can't see on the page. It looks like an ink blot, and that gives me some work for the computation to do, to sort of get used to using the tools. But you're right, it's so simple, it's not the literal solution to anything